Welcome to the Cell Signaling Technology in-house training tutorial for the study of AKT. This tutorial will be broken down into 3 sections. Section 1 of the presentation will cover AKT background information as well as upstream signaling. Section 2 will cover information on downstream signaling, and Section 3 will cover the clinical ramifications of aberrant AKT signaling. AKT is a serine-threonine protein kinase that is a member of the AGC kinase family. AKT, which is also called PKB, was originally discovered as an oncogene within the mouse leukemia virus AKT8, also known as VAKT. AKT, like other members of this kinase family, has an N-terminal plextrin homology domain, also called pH domain, a conserved kinase domain, and a hydrophobic C-terminal regulatory domain, and turn motif. There are 3 isoforms of AKT. AKT1, which is expressed in most cell types, AKT2, which is also expressed in most cell types, and AKT3, which is expressed primarily in neuronal cells. AKT functions to promote cell growth, proliferation, and survival through phosphorylation of a number of downstream targets. These downstream targets contain the AKT recognition or consensus sequence RXR, XXS, or T. In this sequence, phosphorylation occurs on the serine or threonine residue, and arginine residues are required at the -5 and -3 positions. This Western blot shows immunoprecipitation of extracts from serum-starved NIH3T3 cells, untreated or treated with a combination of fetal bovine serum, FBS, and colliculin A, using the phosphoserine-threonine AKT substrate rabbit monoclonal antibody, product number 9614, followed by Western blot analysis using the same antibody. FBS contains large amounts of growth factors that stimulate AKT and other signaling pathways. Colliculin A is a serine-threonine phosphatase inhibitor and is used to maintain the phosphorylated state caused by FBS treatment. With combined FBS colliculin A treatment, a number of phosphorylated bands appear on the Western blot, indicating phosphorylation of proteins containing the AKT recognition sequence. Upstream signaling of AKT. The following is an overview of how AKT signals in the cell. A number of receptors at the cell membrane signal through AKT, including many receptor tyrosine kinases, RTKs, cytokine receptors, G-protein coupled receptors, GPCRs, B-cell receptors, and integrin receptors. These receptors all signal to AKT via a protein kinase called PI3 kinase and affect the phosphorylation state of numerous downstream targets. Now let's look in more detail at how a signal from a cell membrane receptor leads to the activation of AKT, a process called upstream signaling. Membrane receptors signal through phosphoinositide 3 kinase, which is known as PI3K, to activate AKT. PI3K phosphorylates phosphatidylinositol 4,5-bisphosphate, known as PIP2, to generate phosphatidylinositol 3,4,5-triphosphate, also called PIP3. PIP3 binds to the pH domains of several proteins, including AKT and PDK1. This PIP3, which is membrane-anchored, binds to the pH domain of AKT and causes the recruitment of AKT to the cell membrane. The image here shows confocal immunofluorescent analysis, or IFIC, of C2C12 cells that have been treated with either the PI3K inhibitor LY294002 or with insulin using the AKT pan-rabbit monoclonal antibody, product number 4691. AKT pan antibody has been labeled green. Actin filaments have been labeled red with Alexaflor 555 phalloidin. With LY294002 treatment, PI3K inhibition does not produce PIP3, and AKT is not recruited to the membrane. Note the green cytoplasmic staining with LY294002 treatment. Insulin treatment causes PI3K to generate PIP3, which recruits AKT to the membrane. Note the green cluster staining on the membrane with insulin. The AKT pan antibody detects all three isoforms of AKT. At the membrane, PDK1 phosphorylates AKT at threonine 308. Threonine 308 is on the AKT activation loop and is within the AKT kinase domain. This IFIC analysis shows C2C12 cells that have been treated with either insulin or the PI3K inhibitor LY294002 using phospho-AKT threonine 308 rabbit monoclonal antibody, product number 2965, labeled in green. Insulin treatment causes signaling through PI3K, resulting in PDK1 phosphorylation of AKT threonine 308 at the membrane. Note clusters of green-yellow stain at the membrane in the left panel. With LY294002 treatment, PI3K is inhibited and phosphorylation of AKT threonine 308 does not occur. Note loss of green signal in the right panel. PTEN is a phosphatase that converts PIP3 back to PIP2. Therefore, PTEN inactivates AKT and serves as an off switch for the signaling pathway. Loss of PTEN results in constitutive activation of AKT and is often seen in human cancers, an example of which will be shown later in this tutorial. Full activation of AKT requires a second phosphorylation at serine 473 by mTORC2. mTORC2 is the rapamycin-insensitive complex that consists of mTOR, Richter, G-beta-L, also called MLST8, and SYN1. Serine 473 is within the C-terminal regulatory domain of AKT. Seen in this image is IFIC analysis of C2C12 cells, either LY294002 treated or insulin treated, using phospho-AKT serine 473 rabbit monoclonal antibody, product number 4060 in green. Inhibition of PI3K by LY294002 does not cause AKT to be recruited to the membrane and phosphorylated by mTORC2. Note lack of green signal with LY294002 treatment. Insulin treatment results in AKT recruitment to the membrane and phosphorylation by mTORC2 at serine 473. Note cluster of green stain at the membrane in the insulin-treated panel. Other phosphorylation sites of AKT. In addition to threonine 308 and serine 473, AKT is also phosphorylated at other sites along the protein. One of these additional phosphorylation sites is threonine 450. Threonine 450 phosphorylation occurs in AKT's turn motif. The turn motif is a motif also found in PKC and other AGC family members. Threonine 450 has been shown to be phosphorylated by at least two kinases. The first is the mTORC2 complex, whose phosphorylation at threonine 450 promotes proper protein folding and stability in newly synthesized AKT. Threonine 450 can also be phosphorylated by JNK. Phosphorylation by this kinase results in reactivation of AKT in cardiomyocytes after ischemic injury and serves as a priming event for subsequent phosphorylation by PDK1. Here is Western blot analysis of extracts from NIH3T3 cells, either lambda phosphatase or PDGF treated, using phospho-AKT threonine 450 antibody, product number 9267, in the upper panel, or AKT antibody, product number 9272, in the lower panel. Note the appearance of threonine 450 phosphorylation with PDGF treatment. Upstream signaling review. Let's review AKT upstream signaling. Activation of membrane receptors by growth factors, cytokines, or other external stimuli results in signaling through PI3K to generate PIP3. This recruits AKT to the cell membrane, where it is phosphorylated at threonine 308 by PDK1. Then, AKT undergoes a second phosphorylation at serine 473 by mTOR2. Full activation of AKT requires phosphorylation at both sites. The activation state of AKT in cells can be measured using phosphothreonine 308 and phosphoserine 473 specific AKT antibodies. Active AKT phosphorylates many downstream targets to promote cell proliferation, cell migration, insulin action, survival, protein synthesis, neuroprotection, and nitric oxide synthesis. AKT downstream signaling. Now let's take a look at the downstream signaling that takes place once AKT has been phosphorylated at threonine 308 and serine 473. Active AKT phosphorylates a large number of downstream substrates that can be grouped by their effects on protein synthesis, survival, proliferation, glucose metabolism, neuroscience, and other cellular effects. It should be noted that this is a representational diagram and not a complete list of AKT substrates. We will now take a closer look at each of these target groups. Effects on cell proliferation and survival. AKT promotes cell proliferation by inhibiting a number of cell cycle inhibitors. AKT targets in this group include FOXO1, 3, and 4, MYT1, P27-KIP, P21-CYP, and WEE1. IFIC analysis showing inhibition of the transcription factor FOXO1 by AKT in IGROV1 cells is shown here. IGROV1 cells are a human ovarian adenocarcinoma cell line. FOXO1 rabbit monoclonal antibody, product number 2880, has been labeled green. Actin filaments have been labeled red with DY554 phalloidin. Treatment with LY294002 results in PI3K inhibition and inactivation of AKT. FOXO1 remains active and enters the nucleus to activate transcription. Note nuclear localization of green signal with LY294002 treatment. With insulin treatment, AKT becomes active, resulting in the phosphorylation and inhibition of FOXO1. Inhibition of FOXO1 results in nuclear export and inhibition of transcription factor activity. Note cytoplasmic staining of green signal in this panel. AKT also promotes cell survival through phosphorylation and inhibition of a number of pro-apoptotic proteins. Targets in this group include BAD, BAX, BIM, FOXO1, 3, and 4, and MDM2. IFIC analysis was performed showing cellular localization of FOXO3A and SHSY5Y cells. SHSY5Y cells are a neuroblastoma cell line. FOXO3A rabbit monoclonal antibody, product number 2497, has been labeled green. Actin filaments have been labeled red with Alexaflor 555 phalloidin. DNA has been labeled blue with DRAC5. Upon IGF-1 treatment, AKT is active, and FOXO3A is phosphorylated and inhibited, resulting in a cytoplasmic localization of green signal. With LY294002 treatment, PI3K inhibition leads to AKT inactivation, resulting in active FOXO3A that localizes to the nucleus. Effects on protein synthesis. AKT promotes protein synthesis by acting as a major regulator of mTOR. The mammalian target of rapamycin, mTOR, is a serine threonine protein kinase present in two distinct complexes. mTORC1, which consists of mTOR, raptor, and G-beta-L, also called MLST8, is inhibited by rapamycin and functions to promote mRNA translation, transcription, and cell growth when energy conditions are favorable. AKT signaling activates mTORC1 and thus promotes protein synthesis through inhibition of TSC2 and Pras40, both of which downregulate mTOR activity. The second mTOR complex, mTORC2, consists of mTOR, Richter, G-beta-L, and SYN1, and is insensitive to rapamycin. This complex functions upstream of AKT, providing the critical second phosphorylation of AKT at serine 473, which is necessary for AKT activation. Shown here are 3 Western blots depicting AKT as a regulator of mTOR. The Western blot on the left features extracts from serum-starved 293 cells, untreated or EGF-treated, using phospho-mTOR serine 2448 antibody, product number 2971. EGF treatment results in the increased phosphorylation of mTOR at serine 2448. Note levels of total mTOR using mTOR antibody, product number 2972, remain unchanged. EGF signals through the EGF receptor to activate AKT. The middle Western blot analyzes extracts from serum-starved HeLa cells, untreated or treated with insulin or insulin plus phosphatase, using phosphopras-40 threonine 246, rabbit monoclonal antibody, product number 2997. Insulin treatment results in increased phosphorylation of pras-40 at threonine 246. Note levels of total pras-40 using pras-40 antibody, product number 2610, remain unchanged. Also note increased phosphorylation with insulin treatment disappears when also treated with phosphatase. Insulin signals through the insulin receptor to activate AKT. The Western blot to the right shows extracts from NIH3T3 cells, phosphatase or PDGF treated, using phosphotuberin TSC2 threonine 1462 rabbit monoclonal antibody, product number 3617. PDGF treatment results in increased phosphorylation of tuberin TSC2 at threonine 1462. Note levels of total tuberin TSC2 using tuberin TSC2 antibody, product number 3612, remain unchanged. Phosphatase treatment eliminates phosphorylation in the basal state, similar to serum starvation. PDGF signals through the PDGF receptor to activate AKT. AKT and autophagy. mTOR is an important regulator of autophagy induction. Autophagy is the catabolic process that results in the autophagosomic lysosomal degradation of bulk cytoplasmic contents. In other words, during autophagy, the cell uses the lysosome and autophagosome to degrade worn-out, unused, or non-vital organelles or other cellular components. Autophagy is generally activated by conditions of nutrient deprivation, but has also been associated with physiological processes such as development, differentiation, neurodegenerative diseases, infection, and cancer. mTOR is an inhibitor of autophagy. Because of this, PI3K AKT signaling, which activates mTOR, is also an inhibitor of autophagy. Therefore, treatments that inhibit AKT signaling, such as LY294002, promote autophagy. AKT has also been shown to play a role in several neurodegenerative diseases, such as Alzheimer's and Huntington's disease. Some of AKT's targets in the area of neuroscience are Huntington. Huntington's disease is a neurodegenerative disease caused by the accumulation of polyglutamine repeats within the Huntington protein. AKT phosphorylates the Huntington protein at serine 421, which can partially counteract the toxicity that results from the expanded polyglutamine tract. Ataxin 1. Spinocerebellar ataxia 1 is an autosomal dominant neurodegenerative disorder. AKT phosphorylates ataxin 1 at serine 776, which regulates in association with 1433 proteins. This interaction increases ataxin 1 stabilization and accumulation, resulting in enhanced neurodegeneration. GABA A receptor. The A-type gamma-aminobutyric acid receptor is a ligand-gated chloride ion channel that mediates fast synaptic inhibitory transmission. Phosphorylation of this receptor by AKT increases the number of receptors present on the cell's surface, thereby increasing synaptic transmission. And lastly, GSK3-beta. A key downstream target of AKT is glycogen synthase kinase 3-beta, GSK3-beta, an important regulator involved in neuronal development and neurodegenerative disease pathology. Phosphorylation of CRIMP2 by GSK3-beta helps establish neuronal polarity. GSK3-beta is also one of several kinases responsible for tau phosphorylation, and increased GSK3-beta activity is associated with tau neurofibrillary tangles in Alzheimer's disease. This image shows IF analysis of rat hippocampus, untreated or lambda phosphatase treated, using phospho-GSK3-beta serine 9 rabbit monoclonal antibody, product number 9323 in green. DNA has been labeled blue with DRAC5. Note the loss of green signal with phosphatase treatment. In this image, IF analysis was performed in coronal rat brain sections, treated as indicated, using phospho-AKT serine 473 rabbit monoclonal antibody, product number 4060 in green. DNA has been labeled blue with DRAC5. In the striatum section's upper panels, ischemia treatment, which involves surgery where nutrient and blood supply are blocked, results in increased green signal, as compared to the sham treatment, which involves surgery where nutrient and blood supply are maintained. Note the loss of green signal in the ischemia plus phosphatase panel. A similar pattern of expression is seen in the sections of the subventricular zone, as shown in the lower panels. Cardiovascular homeostasis. AKT has a number of downstream targets in other areas as well, for example, in the area of cardiovascular homeostasis. Endothelial nitric oxide synthase, eNOS, is an important enzyme in the cardiovascular system. It catalyzes the production of nitric oxide, a key regulator of blood pressure, vascular remodeling, and angiogenesis. The activity of eNOS is regulated by phosphorylation at multiple sites. Several protein kinases, including AKT, activate eNOS by phosphorylating serine 1177 in response to various stimuli. Western blot analysis of extracts from BAEC cells, phosphatase or VEGF treated using phospho-eNOS serine 1177 rabbit monoclonal antibody, product number 9570, is shown here. BAEC cells are bovine aortic endothelial cells. VEGF signals through the VEGF receptor to activate AKT, resulting in phosphorylation of eNOS at serine 1177. CYP, calf intestinal alkaline phosphatase, is a phosphatase added to reduce background phosphorylation levels, similar to serum starvation. In summary, active AKT phosphorylates a large number of downstream targets that can be grouped based on their effects in the cell, including protein synthesis, survival, proliferation, glucose metabolism, neuroscience, and other cellular effects. AKT can also be found in a number of other signaling pathways, for example, AMPK and insulin receptor signaling, which demonstrates the interconnectedness of many signaling pathways within the cell. As stated earlier, this tutorial includes only a partial list of all known substrates of AKT. For additional AKT targets, please see the AKT substrates table and AKT binding partners table on the web.